Hello, everybody. Welcome back. Sorry, it looks like we had some technical difficulties, but I think we're back online now. So today's webinar, webinar number 1 in our October promotion series, is the keys to soil health. So Adrienne, if you want to go to the next slide. So today's topics, we're going to start with an introduction of our panel. So you get to know a little bit more about us that are going to be talking to you through the webinar. And then we're going to talk about the Soil Food Web training program and the current offer that we have for October. And we'll be introducing a new training program in this offer. And then we'll have an audience poll, and we want to know a little bit more about you and where you're coming from. And so we'll want to get your interaction into this webinar as well. And then we'll get to the really good stuff. This is going to be part of the education that we're going to bring in today's webinar. So part 1 is going to be about what is soil biology. And we're going to talk about the 4 key groups of beneficial soil microorganisms. So we'll introduce you to bacteria and fungi and protozoa and nematodes. And we have a couple short videos that we'll be able to show around that. And hopefully you'll have some questions for us. So we'll talk about how you can submit your questions here in a second, and we'll have a Q&A session to answer some of your questions. And then we'll have part 2, which is going to be how it works. And we're going to be talking about how a balanced soil food web benefits the plants and the growers. And we're going to focus in 2 areas. One's going to be on soil structure. And if we have time, we'll get into how to inhibit pests and diseases. And there'll be some short animations around each one of those. And again, we'll have another Q&A session so we can start answering some questions that you may have. We're expecting that today's webinar is going to take roughly around 120 minutes, so about 2 hours. And next slide. So some rules of engagement that we have for the webinar, just because there's so many people, we have everybody muted for the duration of the webinar, except for the panelists, and that just makes sure that we have good audio quality. And then if you have a question for us as we're going through the webinar, in your screen, you should see a green box, and that's where you'll post your questions. And then we'll select some of those questions for us as panelists to go ahead and review. And if we don't get to your question by today's webinar, then we'll send you an email response within 72 hours. And then also, there's an ability for you to interact with the fellow attendees of the webinar. So there's a purple live chat button on the bottom right-hand side of your screen. And if you click on that, then you'll be able to chat with other folks. And if you need more instructions, you can go and click on the instructions button. And here's an example of what I was talking about. So in the upper right-hand side, you had submitted a question for the panel. And then in the lower right-hand side is that purple button that gives you the live chat option. And we'll be going back to that too when we go to the poll. You'll need to make sure you click on that to open up the live chat, and then there'll be a button for us when we get to the poll. And I'll show you a slide screen of that before we get there. Okay, so introducing today's panelists. You're all here to see and get informed or educated by Dr. Elaine Ingham. So she's the founder of the Soil Food Web School. And then we have Dr. Adrienne Godschlax, and she is also a Soil Food Web researcher and a mentor out of Oregon. Eric unfortunately couldn't make it with us today. If he does join, hopefully you'll be able to see his face and hear him if he's able to join our panel. We also have Wes Sander, who is a certified lab tech, and he's out of California. He owns a company called Foothill Biological. And then myself as a host, I am Brian Vagg. I am a soil food web consultant, and I own a company called Sprouting Soil, and I'm based out of California as well. Okay, so that's our panelists. So we want to talk about the promotion that we're running in October. This is going to run through October 21st. And we're introducing a new educational track. It's going to be the Growers Training Program. So that's represented on the left-hand side. And this is going to be continuing education for farmers, ranchers, and growers. So these are folks that don't necessarily want to be a consultant, but they want to know more information about how to implement these practices on their specific farms. And then on the right-hand side, we have the Consultants Kickstarter or the Consultants Training Program. And this is where you can become a consultant, run your own lab, be a biological compost producer. And what's nice about the promotion, this is the biggest promotion that we've run so far. Typically, each one of these tracks run around $7,000. For the duration of this promotion, we're reducing the price by 45%. And so that's about a savings of $3,170. So that's very, very significant. In fact, it's the biggest discount we've offered so far. And if you want to know more about these programs, on the bottom of your screen, there will be a button that you should be able to click, and it will bring you to how you can sign up for these different programs and get more information. Now, we're also going to show you a video. We're going to have two videos. The first one's going to be talking about the growers training program. And then after this, we'll go to a second one that will talk about the consultants training program. So Adrienne, if you want to kick that off.
This October, we're presenting our biggest bundle ever with the biggest savings we've ever offered. With the BioComplete Compost Bundle, you can register for the Soil Food Web Foundation courses with Dr. Elaine Ingham for just $38.70, saving over $1,100 through October 21st. You'll also get Stage 1 of the Grower Training Program totally free, saving a further $15.40. That's 26 hours of mentor time dedicated to helping you make your own biological compost and develop your microscopy skills to the standard required to qualify as a certified soil food web lab technician. You'll also get 2 free bonuses with this offer: the Introduction to Permaculture course by Graham Bell and the all-new Soil Sponge Regeneration workshop with Dee Dee Purshouse, saving a further $500. This is the biggest discount bundle that we've ever offered, with a total value of over $7,000, for which you'll only pay $38.70, saving you a total of $31.70. That's 45% off. In Foundation Course 1, you'll take a deep dive into the science and methodology that underpins the soil food web approach, which was developed by Dr. Elaine Ingham over the last 4 decades. You'll study the history of soil on planet Earth and how the agricultural practices we've been using in the last 100 years have degraded our soils to the point where we now only have around 60 harvests left, according to the United Nations. You'll learn about the solution to many of the problems that are familiar to farmers all over the world today. Problems like diminishing soil fertility, pest and disease pressure, low crop yields, drought, flooding, compaction, and soil erosion. Regenerative agriculture can address all of these problems. You'll be introduced to regenerative practices like no-till and the use of cover plants. You'll also learn about the 4 major groups of microorganisms that drive soil regeneration and how the process can be accelerated by restoring the microbial community or soil food web to your soils. Dr. Elaine will present a number of case studies from around the world that she has worked on personally. Here you'll see some of the amazing results that have been achieved using the soil food web approach.
In Foundation Course 2, you will learn all about the importance of biological compost and how it's very different to regular compost. When most people look at compost, they only see a means of delivering nutrients to their plants, so they think about how much nitrogen, potassium, phosphorus, and other elements are in the compost. When we look at biological compost, we see a means of inoculating the soil with beneficial microorganisms so that the soil ecosystem can start to function again, providing plants with a continual supply of nutrients. This is kind of like teaching someone to fish so they can feed themselves for life, as opposed to just giving them a single meal. In this part of the training, you will learn how to make biological compost using various starting materials to create a recipe that will produce results. You'll learn how to monitor and control moisture levels, aeration, and temperature in order to ensure that beneficial microorganisms are multiplied, while disease-causing organisms are destroyed or become dormant. You'll also learn about the various types of equipment that can be used at different scales, and you'll learn about the different ways in which biological compost can be applied to the soil. In Foundation Course 3, you'll study Dr. Elaine's methods for making liquid biological soil amendments. Compost extracts are used as a soil drench, delivering microbes deep into the soil profile, where they can interact with the plant's root system. Compost teas are applied to the plant's foliage, where they form a protective barrier against foliar diseases. In time, as your soil biome becomes more diverse and vigorous, your plants and trees may be entirely covered with beneficial microbes without having to continually apply them.
In Foundation Course 4, you'll focus on 4 major groups of microorganisms in the soil food web: bacteria, fungi, protozoa, and nematodes. You'll learn how to use a compound microscope to identify and quantify these microbes so that you can really see what's going on in your compost and in the soil. This will give you the ability to assess the quality of your compost and liquid amendments before you invest all the time and effort that is required to apply them to the soil. You will also be able to monitor the progress of the microbial community in your soil over time, so if something is going wrong, you will know about it before the plants start to suffer. This gives you the opportunity to take remedial action early on in the growing season. Next, in Stage 1 of the Grower Training Program, you'll be assigned to one of our highly skilled mentors. some of whom are soil food web consultants who run their own businesses supporting farmers to make the transition to the soil food web approach. And others are PhD biologists who have been working closely with Dr. Ingham for several years. You'll work one-on-one with your mentor to develop your compost making and microscopy skills. When it comes to making great biological compost, there are many variables and every situation is different. Our mentors have worked with dozens of students making compost in many different conditions, so they'll be able to help you address some of these challenges that are unique to your location. They'll also guide you to avoid making common mistakes that can cost you lots of time, money, and effort. Your mentor will support you as you develop your skills to the required standard to pass the microscopy proficiency assessment. Achieving this standard will give you the confidence to really know what's happening in the microbial community in your compost and soil. You'll be able to perform an assessment of your soil in about an hour or less, so you won't need to rely on an external lab. Once you successfully pass the microscopy proficiency assessment, you can elect to be listed on our website as a certified soil food web lab tech, which means that you can assess soil samples for other farmers and growers in your region.
You'll have a total of 26 hours of mentoring time, so you can arrange Zoom calls and exchange emails with your mentor whenever you feel you need support. Our mentors work together as a team, and there's a lot of diverse experience and knowledge between them, so you'll have access to a great deal of support. You'll also have access to the 2 bonus courses. In the Introduction to Permaculture course, you'll learn from internationally respected teacher, author, and lecturer in permaculture, Graham Bell. This is a series of 18 lectures and quizzes where you will take a deep dive into this amazing design system that can be applied when designing your home, garden, farm, business, or community. Permaculture is all about being in balance with the planet and is an ideal tool to have in your bag whenever you're considering making a change. The Soil Sponge Regeneration Workshop is delivered by educator and author Dee Pershaus. This 5-session course is all about regenerating the soil sponge for flood, drought, and wildfire resilience. It builds on the successes of innovative land managers around the world who are saving huge sums in damages from extreme weather events and crop diseases while restoring the dignity and profitability of farming. Dee teaches participatory workshops both in person and online, helping to show the nested relationships between soil health, human health, water cycles, and climate resiliency.
So just to recap, the BioComplete Compost Bundle comprises Soil Food Web Foundation courses with Dr. Elaine Ingham, where you get to study the theory and application methods behind the Soil Food Web approach, and that all-important 26 hours of mentor time in Stage 1 of the Grower Training Program designed to help you develop your compost making and microscopy skills. You'll also get the 2 free bonuses, Introduction to Permaculture course with Graham Bell and the Soil Sponge Regeneration Workshop with Dee Pershaus. This bundle is ideal if you want to make your own biological compost and master the microscope. These are the skills that are most in demand amongst farmers who come to the Soil Food Web School. Once you have completed the foundation courses in Stage 1 of the Grower Training Program, you'll be well on your way to improved soil fertility, greater yields, and resilience to pests, diseases, drought, and flooding.
Right. Now, that was talking about the Growers Training Program. We're going to talk about the Consultants Training Program, and then we'll be moving on into our educational track.
This October, we're presenting our biggest bundle ever, with the biggest savings we've ever offered. With the Consultant Kickstarter bundle, you can register for the Soil Food Web Foundation courses with Dr. Elaine Ingham for just $38.70. Saving over $1,100 through October 21st. You'll also get Stage 1 of the Consultant Training Program totally free, saving a further $15.40. That's 26 hours of mentor time dedicated to helping you make your own biological compost and develop your microscopy skills to the standard required to qualify as a certified soil food web lab technician. You'll also get 2 free bonuses with this offer, the Introduction to Permaculture course by Graham Bell and the all-new Soil Sponge Regeneration Workshop with Dee Pershaus, saving a further $500. This is the biggest discount bundle that we've ever offered, with a total value of over $7,000, for which you'll only pay $38.70, saving you a total of $31.70. That's 45% off.
Is that the whole video?
All right. Yep, that was it. Nope,
Okay.
I think that was it. Okay, so it was great to be able to see those videos explaining the different promotions that we're running for October. And again, remember that's through October 21st. And I think, when you go to the next slide, there you go. Give us 1 second here while we
I'm locked into permanent logo mode.
just gonna watch the promotion for the rest of the webinar.
All right, hold on. Let me try to get out of here.
Sure.
Okay. I think we're still here.
All
Yay!
right.
We can
Good deal.
continue.
Okay.
Thanks, Brian.
So I also want to talk about some of the upcoming webinars that we have coming up, for the rest of the month. So we have another webinar, webinar number 2, that's going to be proof that regenerative ag works, and we're going to have a guest speaker there as well, and Nicholas Netian. We're really excited to be able to have him coming in. He's done some fantastic work around olives. He's made some olive oil that's just phenomenal, kind of world renowned with the polyphenols that are found in his olives. And then we have webinar 3, which is going to be how to rapidly regenerate your soil. And that's going to be on Thursday, October 21st. And so we're going to be talking about how to inoculate your soil with beneficial microbes, and really how what the practical applications are in doing that kind of work. And then we'll have webinar 4, which is going to be on Thursday, October 28th. And that's going to be Meet the Soil Regen Pros. So you're going to be able to hear from a number of us that are actually working out in the field and making those transformations. How are we going about in transforming the agricultural and landscapes and all that kind of good stuff. So really exciting series of webinars coming up. Okay, we can go next slide. All right, so before we get into the educational portion of this webinar, we do want to hear a little bit from you. So you can go to the next slide, Adrienne. And we're going to launch a poll, and we're basically going to be asking you kind of what best describes where you're coming from. Are you coming to this webinar from a farmer or ag professional capacity, or are you coming from more of an environmental advocate? And we really just use this information to help us tailor to say, hey, are we reaching the right folks? So we'll spend maybe 20-30 seconds running the poll. And again, you'll have to click on the purple chat button to open up the dialog box, and then you can see the arrow on the right hand side. There's a little kind of button that you'll be able to press to be able to put in your poll answers. So fantastic. Thank you everybody for participating in our poll. Okay, I say let's move on to the next part of this, and we're going to get into part 1 of our webinar educational track, and it's going to be around what is soil biology. And so we're going to watch a short animation on the soil food web, and then we're going to have Dr. Elaine talk about some of the different organisms, and we're going to watch some videos of some of the different organisms that you would end up seeing when you're looking through a microscope. So let's go ahead and kick off that animation.
What is the soil food web? To understand the soil food web, let's start with the term food web. We all know about the food chain, the animal kingdom on top of which are us humans, right? Well, if we look a little closer, we can see that some members of the food chain don't just eat one thing. That goes for humans too. So the reality is more like a web than a chain. This is the food web. There happens to be a food web in the soil too. This is the living part of the soil made up of insects, earthworms, and much smaller microscopic creatures such as fungi and bacteria. Dr. Elaine Ingham has pioneered research into the microorganisms in the soil over the last 4 decades, and has worked with a team of research scientists to understand how they interact with each other and with plants. The soil food web can be thought of as the soil biome. Just as humans have a gut biome responsible for digesting our foods, so too the soil has a biome, which breaks down organic matter and releases nutrients in plant-available form. This is how nature has been feeding plants for billions of years. The major groups that make up the soil food web are bacteria, fungi, protozoa, and nematodes. When in balance, these different groups interact with each other and with plants to create abundant ecosystems such as the great forests of the world. Have you ever wondered how forests can be the most productive ecosystems in the world without the need for any fertilizers or pesticides? The answer lies in soil biology. With a healthy biome, the soil can provide plants with all the nutrients they need, and with a number of other benefits, such as protection from pests and diseases, protection from drought and from flooding. The soil food web is, essentially, nature's operating system. Unfortunately, we humans have disturbed the soil food web in almost all of the soils that we manage, causing it to become unbalanced. As a result, the plants we grow struggle. Plowing is the major cause of the problem, as it destroys the larger microorganisms, such as fungi and protozoa, leaving the soil food web out of balance. This results in a system breakdown. Nutrients are no longer made available to plants, and protection from diseases is compromised. Before the Industrial Revolution, humans would plow using oxen or a bull, which provided around 3 or 4 horsepower. Modern tractors can yield 400 horsepower or more. So far more damage is done to the soil biome by modern machinery. The use of chemicals has compounded the problem. The good news is that we can restore the soil food web to most soils within just a few months. This results in a number of benefits, both for farmers and for the environment. With a balanced soil food web in place, farmers need not use fertilizers at all. They don't need to use pesticides either, as nature's operating system protects plants from attack. Herbicides, used to kill weeds, are not required either, as weeds only thrive in conditions where the food web is out of balance. Restoring the soil food web means farmers save money on chemical inputs across the board. It also means that their yields increase dramatically. In some cases, farmers working with Dr. Elaine Ingham have seen yields increase by over 200%. This is because the soil food web provides plants access to a constant flow of nutrients from soil organic matter, and from the soil particles themselves. That's right, sand particles contain nutrients, and guess what? Fungi and bacteria can harvest those nutrients. They then make these available to the plant in a process that the plant actually controls. This means that plants get access to the type of nutrients they need precisely when they need them. That's how you maximize yields and optimize profits. For the environment, there is a whole host of benefits of having a balanced soil food web. Humanity is facing a number of existential threats. Let's take a look at how some of these are related to the soil. The most obvious one is soil erosion. The United Nations Food and Agriculture Organization has estimated that we only have around 60 years left before all the world's topsoil is depleted. Other estimates are as low as 30 years. The soil food web prevents soil erosion by both wind and water by building structure. Please watch the animation on soil structure for more information. Another existential threat we are facing is ecosystem collapse. The UN has recently stated that insect populations have been decimated by around 25% each decade for the last 30 years. That means that there are 57% less insects today than there were in 1989. Bird populations have declined by 1/3 in the last 15 years in parts of Europe. So how does the soil food web help? Well, the UN has identified the use of pesticides as a major cause in the decline of insect populations. Please watch the animation on pests and diseases to find out how nature's operating system protects plants against attack, eliminating the need for pesticides. Another threat to life on Earth is climate change. Fortunately, the soil is capable of holding tremendous amounts of carbon in the bodies of microorganisms, and some mega-organisms too. The biggest living organism in the world is not a whale, it's a fungus found in Oregon that is the size of 1,665 football fields. It is between 2,000 and 8,000 years old, and it is made mostly of carbon. By restoring the soil food web, we could put a stop to climate change. Please watch the animation on soil carbon sequestration for more details. At the Soil Food Web School, we train people like you to help farmers transition away from using chemicals to farming in harmony with nature by restoring the soil food web. For more information about the numerous benefits of the soil food web and how you can get involved, visit .com.
All right. I always love watching that animation. So well done. Okay, we're going to actually hand this over to Elaine, and Elaine is going to talk to you about the bacteria, the fungi, the protozoa, and the nematodes, those 4 key microorganisms that were mentioned in the video. So, Elaine, take it
Oh,
away.
thank you, Brian. So I'm going to briefly discuss these 4 groups of the soil organisms that you need to have present in your soil. Probably the first thing I want to talk about is the fact that it's the balance of these organisms that determine which kinds of plants are going to be promoted. They're going to get all the nutrients they need from the soil without compromising your above ground parts of your plant. So quickly, fungi and bacteria both decompose organic matter, but bacteria go after the simpler, easier compounds. Bacteria can only make one enzyme at a time. And so it's got to pick off all of the material on the outside that is that bond to break down that organic matter. Then it can make a different enzyme and break down the next. Well, you can see kind of how that can get to be pretty slow. So bacteria are going to go after the easy to decompose things, so this happens as rapidly as possible. While fungi are going to decompose the more complex materials, where they've got to be making several enzymes at a time to key into all the diversity of the different kinds of bonds in that organic matter to be able to break that down. Bacteria, when they consume organic matter and break down any kind of organic matter, dead bodies included, bacteria have to concentrate all of the nitrogen, phosphorus, sulfur, magnesium, calcium, etc., that's in that plant material. And so the bacteria have to exit all the excess carbon that's in the food that the bacteria are eating. And therefore, they're concentrating nutrients, but how do they get rid of that excess carbon? They blow it off as CO2. When we're dealing with fungi are decomposing that complex material, even huger amounts, larger concentrations of carbon in that material, but fungi don't release it as CO2 into the atmosphere. The fungi take that carbon and use it to reinforce their hyphae, the strands through which nutrients have to come to that fungus. So that fungus is using that carbon for something very important. And that means not that much of the carbon that they are getting from their decomposition is going to be blown off as CO2. So you can see some of the really important functions of bacteria and fungi. Building soil structure is another one. How do you take each of the individual sand, silt, and clay grains and try to stick them together so that they will open up more air passageways, more hallways to allow oxygen and water and your roots and these organisms to go deeper and deeper into the soil? You need to build soil structure. And the first step is done by making microaggregates, which is what bacteria do. They make the glues to bind those aggregates together. When we're dealing with aerobic bacteria, they're going to be alkaline in nature. If you're looking at anaerobic bacteria, then very low pH, highly acidic, will harm plants almost across the board if you get those anaerobic bacteria growing. So we need to build soil structure and maintain oxygen concentrations in the soil. The bacteria start that process. Fungi come along then and they bind with their strands of hyphae, and you can see a strand of hyphae here in the picture next to it, strand of fungal hyphae, and they bind all those microaggregates together, building larger pore spaces, building larger airways and passageways for the water, for nutrients, for the microorganisms, air, to move deeper and deeper into your soil. If you've ever had a problem with compaction, you know how deadly that is, because as soon as you get any water presence sitting on the surface of that compacted layer, the microorganisms in that puddle basically are going to use up all that oxygen, and now you go anaerobic, and that pH is going to kill your roots. It's going to kill the beneficials. So we want to make certain that we keep things aerobic. Retaining nutrients in the soil is very important. We have predators then that eat the bacteria and fungi, because the bacterium, and that's very important, because the plants have to get the nutrients that are bound or tied up in the bacterial and fungal biomass. So the plant is putting out exudates to tell the bacteria and fungi what enzymes to make, to go and pull those enzymes out of the organic matter and also out of the silica bilayers in your sands, your silts, your clays, your rocks, your pebbles, your parent material. You will never run out of nutrients made available for your plants as long as you've got the bacteria and fungi pulling those nutrients into their bodies. But now it's like those nutrients are tied up in a pantry. How does the plant get it? That's where you have to have the nematodes and the protozoa come along and eat their favorite groups of organisms to consume. And when that happens, the concentration of those nutrients in the bacteria and the fungi are so much higher than what the protozoa or the nematodes need. So the nematodes, the protozoa, what are they going to do with all these excess nutrients? Because it's going to kill them if they try to keep those inside their body. There's just too high concentration. And so they release those out into the soil in plant available forms right next to the root system of your plant, because of course the bacteria and fungi being fed with the exudates coming from the plant, that's where all of this cycling process is going to happen then, is right next to the plant. So the plant can just take the nutrients it needs immediately, right on call. Every second of every day, your plant is being fed the proper balance of nutrients that that plant requires, if and only if you have these organisms in the proper balances for your plants. So, quick overview of some of the extremely important things that go on in the soil that probably you've never heard about before, because this is pretty new data, scientifically speaking. But we've looked at soils all over the world. We've looked at every major crop type all over the world. And our overarching principles remain the same. Everywhere we go, these same interactions have to be occurring. So you don't have to be putting down inorganic fertilizers or pesticides or herbicides or nematicides or all those acidal things, which of course kill these beneficial organisms, as well as maybe kill some of the bad guys. Okay?
Yeah.
Oh, back over to you, Brian.
Oh yeah, thanks, Elaine. Yeah, it's always good to get that brief introduction into the soil food web. And in the courses, we definitely do a much deeper dive in describing each one of these functions about fungi, bacteria, and predators of the system. But we also wanted to share with you, just so you can see some of these organisms. It'd be, if we were talking about farming cattle, I think everybody's probably seen a cow before, or a pig, or some kind of animal. But most of you probably have never seen a nematode or a protozoa or a fungi or a bacteria. And so we use a tool, which is the microscope. And here are some videos. We're going to be looking at some of the different organisms. So we're looking at a testate amoeba that's going to appear here in the screen. And remember, they are predators. They consume bacteria, and they're going to release nutrients in a plant available form.
And they like particular bacteria. So you can see where those pseudopods are kind of, oh, no, I don't want to eat that. And then going on to grab some other bacterium.
It's just neat how it just creeps in through the soil profile, just gathering bacteria and just pulling it into its body.
And releasing nutrients for your plants.
Yeah, this is a beautiful video. I just obsess over these organisms. I think testate maybe are unsung heroes. And you can really see the organism itself is kind of like a snail, if you will, where there's a shell and then it's an organism that extends out from within that shell. So that's what we're looking at there. And right at the end of this video, we get a little flagellate that comes up from the bottom here too, another protozoan. Although it goes fast.
Yeah, totally different type of protozoa. But yeah, it's fantastic. You know, it's funny when you first start becoming a lab practitioner, you start really looking at organisms in the soil. Boy, you could just sometimes I get so distracted. It's like I have a job to do. I need to get the soil, sample done. And ooh, what's this organism? I've never seen that before. And pretty soon you're half hour into just watching that one organism, going, okay.
Distracted by shiny objects.
Complete. And there's so many new shiny objects to look. I mean, I'm amazed. I've done thousands of soil samples, and I constantly see new things I've never seen before.
I'm
All right,
constantly seeing new things, and I've been doing this for the last 45 years.
Exactly.
There's always something new. Nature in her gorgeous, incredible diversity.
So here we're looking at fungal hyphae and some plant matter that's going to be in here. And now remember, again, fungi, they're decompose organic matter. And again, they're going to have the enzymes to extract those nutrients from the organic matter, the sand, silts, and clays. And again, they're going to hold nutrients in their bodies, so they have to be predated upon as well. And so they're going to be organisms that are going to eat fungi. And they also have other roles about creating the macroaggregates, creating that soil structure by tying those microaggregates together.
You can see over on the left-hand side where these fungi have started to work, tying all these little micro aggregates together in this larger macro aggregate that's the fungal hyphae is going to pull it all together. There's all kinds of cute little critters running around at the beginning of that. And I hope you noticed them. Some were flagellates and some were bacteria, motile bacteria.
Okay, I have to give a shout out here because this is one of my students' videos, and I'm very proud of her. Way to go, Johanna. So this is a nematode, and this is the predator of that last image. They would take this spear that you can see that's rigid right here in that mouth part, and they would pierce that into and break open that the chitin and that cell wall of the fungus, and suck out all of those beautiful, delicious, juicy contents.
They have a built-in straw.
Yep. And they got it. And so very important in releasing nutrients in a plant available form right here in the root system. So your plant's going to be fed every second of every day with the proper balances of the nutrients they need. There is no soil on this planet that lacks the nutrients to grow plants. What we've done is to destroy the microorganisms in your soil. So with the toxic chemicals, with the constant tillage, with the pesticides, herbicides, all of those things, and you don't have the organisms left for them to do the change that has to happen to go from the total nutrients tied up in the sand, silt, clays, rocks, and pebbles in organic matter and turn that into a plant available form. So that explains why you can put inorganic fertilizers down, you get a big increase in growth. The nutrients are there in your soil. Why are you adding something you don't need? Because we've killed off the food web.
Here's a fun one. And so there's some other organisms, this one we're looking at two organisms, tardigrade. Common name for this is the water bear. And it's the one on the right-hand side, looks like it has the paws kind of just going up. Well, and there's a rotifer that's going around and basically, I think, cleaning out the outside of that tardigrade. Tardigrade's getting a car wash.
Yeah, it's like those cleaner fish that sharks have, they go along and chow down and keep the other guy all nice and clean and tidy.
It's like he's tickling him.
It is kind of like tickling. He's like, oh, come on, get the spot over here.
Yeah, I was like, ooh, that feels good.
And here we got another one where we're looking at ciliates. And ciliates are another part of the protozoa. Ciliates are fantastic to watch. We call them race car drivers of the soil, but they also give us some other ideas. When you see large quantities of ciliates, they tend to thrive in anaerobic conditions. So if we're starting to see a lot of ciliates, that's a clue for us to say, we need to investigate this more.
Something's going wrong here.
Do we have some flagellates in here too, Brian, do you think?
I think so, but they're moving so fast.
Smaller ones.
The smaller ones, yeah.
Yeah, exactly.
Mm-hmm.
Not too many, though. Way
the really
more.
Small ones. There are these small ciliates, and then the really small ones down
small
in there.
Ones, yeah.
Right in there, yeah.
Middle of the screen, exactly.
Mm-hmm.
This is when you start seeing kind of an imbalance. Typically, if we see a couple ciliates, it's not a big deal. In fact, ciliates will tend to live in earthworm or worm casts. And so a few, that's not so big of a deal. But when you start seeing a lot more ciliates as the dominant protozoa, then that's when we start going, okay, maybe we've got some issues with the soil. And that's a neat thing about learning these things. The organisms that we see in the microscope are very good indicators to us. What is the conditions of the soil? Are we moving in, or are we in an aerobic condition? Are we in an anaerobic condition? Or are we in a kind of a facultative in between those two places?
If I had a band, I would name it Ciliate City.
That's a great name. Okay, I see that we've got quite a few questions around this part, and so I'm going to tee one up for the panelists here. And this is something I think that's been kind of in the zeitgeist. It's very present, is around fire. And so the question is from Lisa, and Lisa asks, is it true when soils have intense fire, i.e. bushfire, on top of it, it bakes and kills the soil for a very long time / forever ?
Well, it's not going to harm the soil forever because the organisms will move back into the areas that have been burned. It's really a question of how fast and who's going to come first. If it's just been a light fire that burns over the surface, it doesn't really raise the temperature of the soil itself, there's very little damage to the organisms in that soil. So they're going to start promoting, those microorganisms in the soil will promote a later stage of succession. And those will be the seeds that will be germinated. Those will be the ones that will get going. And so you sort of jump into the mid grassland kind of condition. You get maybe a few weeds, but not many. Now, if that fire goes across the surface of the soil and it starts burning the organic matter, there's enough thermal load there that what is in the top layers of the soil, the organic matter starts to burn. Now you're in trouble because that gets hot enough, long enough, and almost all the organisms will be destroyed. And so you're going to go back to bare dirt where you don't have any good guy organisms left. You have maybe a few bacteria. And rebuilding from that place is very difficult. I know places up in the Cascade Mountains in Oregon where they had a burn like that back 70 years ago, and it's still in weed stage today, 70 years later. They have not managed to, the weeds haven't managed to accumulate enough organic material in the soil to move that property into a later stage of succession. It's still waiting. But what does Mother Nature care? So there was fire here. It'll get, don't worry, it will become old growth forest one day. She'll get you there. But we can jumpstart these kinds of situations. We don't have to wait for Mother Nature. We have to play within her rules. So we're not messing things up and making things go downhill even faster. We want to get that biology back into the soil, get the organic matter, get the plants growing. So there are things that we will teach you to jumpstart that successional process.
Yeah, I always look at it, for me, there's kind of two parts of this. One is if we have such a bad fire that it, like you said, it burns it back to primary succession. It's such a bad fire, it just kills everything. And we have a role, we can go in there and help accelerate that. But I think on the second part of that is we also can become better forest managers. A lot of these forests already have a fire ecology. That's what they've evolved to be. And by prescribing burning, keeping the fires cooler, I think we can then really protect the organisms in the soil, as well as make that ecological niche really thrive.
You burn the property when it's wet enough outside that it's not going to spread far if it does get out of control for a short period of time. We've been, in a number of our consultants, they've been putting really good biology out, getting structure back into the soil so they don't have to irrigate in the late summer time period. The plants pretty much are taking care of themselves. And then when the wildfire comes, and burns all of the houses all around them, their house is still there standing and none of their plants have burned because there was plenty of water. We need to investigate that, but it is, several times now that we've seen that relationship where one of our consultants had their property was in the path of the fire and it didn't burn. So you can see why that would work, but how do you demonstrate that scientifically?
We're gonna volunteer.
Yeah, fire is a really useful, interesting way for us to a context with which we can understand how we interact with nature, and we can have a beneficial interaction with nature that is not a lack of interaction with nature, because there was that pristine myth that when white settlers came to the US, there was no management of forests because they were huge and lush, and yet they were intensively managed, and fire was a part of that tradition. And so I think there's a lot of ways that we can have a beneficial interaction with nature and learn from some of the communities that have more wisdom there too. And soil biology can be a part of that, and that's really exciting.
Okay, any final comments before we move on to another question?
Head to another question.
Okay, so here's a question anonymously submitted, but the question is, I learned long ago that a teaspoonful of healthy soil contains about 1 billion microorganisms. How many of these life forms have been identified? I think this is a great question.
I think we're probably somewhere at under 1%. Because when you think about all the bacteria, all the different species of fungi, protozoa, nematodes, microarthropods, earthworms, incatraids, all of those critters in the soil, we've barely begun to figure out their taxonomy and what is it that each individual species actually does under varying conditions. We know that as climate change happens, we need to be moving the biology that's in this ecosystem. As this ecosystem moves northward, presumably, we've got to make sure that all the microorganisms and the other animals in that system move along, as well as the trees and the shrubs and the grasses and the forbs. So, and we haven't even identified it, probably less than 1%. Berge's Manual of Determinative Bacteriology lists all the known species that we have named. And in that book, there is 5,000 species that are named. And there are probably 1 billion species of bacteria on this planet. What we don't know.
I mean, there are people that, their entire professional career is a small niche of a particular type of organism, just identify the species, right? and there's a lot of different species that are out there.
So we do take a kind of a different attitude about, do we have to identify to genus and species? We identify to function. And that's, I think, what is most important. And that's what allows us to more rapidly be able to deal with these systems. We're talking about community and ecosystem levels. We're not talking about genus and species. Sorry, Wes, I interrupted you.
Oh yeah, no, not at all. I was just thinking it's a good perspective on this to just consider the microbial inoculation products that are out there on the market. You look at look at a handful of those, and they all kind of have the same species. It's a relatively small number, like a product with a lot of species, and it might have like a dozen. Others have 1 or 2, and they're all the same species. Just and this is like what Brian was saying. Researchers tend to spend their time on one species or something like that, and usually it's concentrated on these ones that go into producing products. And so that's kind of where soil microbiology is most of the time has been spent on this small number of useful species out there. And that's kind of the bigger picture of where we're at with all this at the moment.
And it needs to change.
Yeah, for sure. This kind of reductionist thought of, just a couple of these are going to do wonders. Some people see results from, but it's really the diversity of microorganisms that's the key. is really the key to unlocking a very healthy, vigorous ecosystem.
Yeah, and I would add to that as well that, one of the parts of what we do is we think about ways to grow a diversity of microbes that we know can thrive in those oxygenated, healthy soils. Whereas there are also a diversity of microbes out there that can consume plants as a food source, and we're trying not to grow those and get those out of the field. So we are careful and working towards making sure that our methods ensure a healthy community, a healthy soil food web based on community ecology principles that allows us to block out and prevent those plants from being attacked by pests and pathogens, but we'll get there even shortly.
Because when we start invoking pests and pathogens, one of the primary things we have to do is get the proper nutrients and the proper balances back into those plants. From the day they germinate and start to grow, we want to make certain that the right biology is feeding that plant every nutrient it requires every second of every day, all night long, not just during daylight hours. And so we do want to get that whole diversity back into the soil, because a plant that has all the nutrients that it requires is much more resistant. It is capable of preventing disease-causing organisms to get to any of the plant tissues. And then we want to coat all of the outside layers of those plants that we care about. We want them protected by a deep layer of bacteria and fungi, protozoa, and nematodes. They'll do nutrient cycling on the leaves, eating the bacteria and the fungi and growing more protozoa and more nematodes. So you get more nutrient cycling going on the leaves themselves. And when a disease-causing spore comes along and lands on that leaf, it doesn't land on the leaf. It lands on the bacteria, fungi, protozoa, and nematodes, and here's this nematode that would like to eat it. So we deal with the diseases and pests from all of those different approaches.
Yeah, and a punchline to that is that not all compost is compost in the way that we see it, as vessels covered in the helpful microorganisms. There are composts sold out there that contain pathogens. There are composts out there that don't have a whole lot of life that are called compost. So we try to break down that term. So we use what we call biocomplete compost. There's a lot of other terms out there that can inform. It's the amount of organisms that were carefully grown in that compost. That is the key part.
And that's so true. I can hands down, just through my experiences, that the vast majority of commercially produced compost is not anywhere close to biologically complete. Some are better than others, but they're typically missing massive, portions of the soil food web, where when we make compost, or producers that are making really good compost, you see all the microorganisms they're hoping to see in a compost. That's one of the biggest changes I think that needs to be done in the area of agriculture, is making better compost.
Absolutely. Okay. Most of the compost out there on the market is definitely not what you want. And some of it can be truly detrimental. I've seen whole crops lost when they put out anaerobic, stinky, smelly black stuff that they were told was compost. Nope, that is not compost. Don't buy it.
Yep. Okay, I say we move to another question, but did you have one last comment to make there?
Yeah, one last comment. You want to buy the compost that smells like forest soil, that is brown, deep, rich, 70% cocoa chocolate color brown. And you want to, when you lift it up, you want to shake it a little bit, and you want to see airways and passageways moving into that compost so that you know that it's aerobic. Okay, now I'm done.
Okay, no problem. All right, so we have a question from Justin, and Justin says, I live in Oregon with local access to established forests. Are there any concerns with inoculating my permaculture garden with soil microbes, and more specifically, fungi found in said forests?
I always like to make certain that you're killing any pathogens that might be hiding out in that forest. So I always want to bring that material as an inoculum and put it into your biocomplete compost that you're making. And, we teach you how to do that. So, bring the inoculum, but there might be diseases, there might be pests, there might be problems in that, especially with all the horrible things us human beings have been doing to those kinds of situations. Even though you think you're off miles and miles away from any other person, they've been logging there in the last 20, 30 years. Look at the size of the diameter of the trunks at the base. If it doesn't take 3 people to stand around it, something's been logged. And so what are the diseases and pests that they brought in? Has this area dealt with the diseases and pests, or might you not be bringing something deleterious if you just pick it up out in the forest and then go apply it to your beds? You could be bringing some real problems in with you. So we always encourage you to compost it correctly, where we know that we have gotten rid of all the diseases and pests. We look at the compost using our microscope and making certain that we have only beneficials in that material.
Yeah, I did what you said. I mean, really, it's a common practice for us to go out and take inoculum from the surrounding. We want to make our compost that we're creating to have the indigenous microorganisms. But like Elaine said, we want to make sure that we're knocking out any kind of pathogens through a heat cycle, and thermophilic composting is the way to go. And so, yeah, very common for me. I'll walk around. I'll go on special trips to go hiking in different areas, go collect up materials when I'm making a new compost pile, just so I can get some new diverse set of indigenous microorganisms into that pile.
You will never again go on, take time off and go on vacation, right? Instead, you're going on a organism collecting trip. You want more diversity, and so you're collecting, and your business should have to pay you for that.
I have a backpack that we typically want to go on hikes, and for the last 4 or 5 years, there's always baggies in that that backpack, no matter where I go, just in case. Oh, that's interesting.
Just in case.
Exactly. Adrienne, Wes, anything else you guys want to add?
Oh, just that's a really cool and such a big part of this whole journey is to build up your microbe stocks. And yeah, you just go around collecting them wherever. And it's so cool to watch your compost become more rich over time as you keep collecting things. Some things were new to me when they first showed up in my compost, a year or two ago, perhaps. And now I see them all the time. They're always there.
Yeah.
Mm-hmm.
It's just, it's such a cool thing to see.
I will also advocate as an ecologist the role of your local microbial community. Oftentimes certain microbes are widespread, but other microbes are endemic to only one spot. And the best rule of thumb is that your microbes always know best. Your local community, they know how to deal with your typical heating regimes throughout the year, the amount of rainfall, so they're used to those normal habitat conditions. And so the more that you can not exploit, but collaborate with your local microbes in your habitats, the better your compost.
Exactly. Okay, I say we conclude part 1 and we'll move into part 2. So part 2 is we're going to be talking about soil structure to start with, and then we're going to get into inhibiting pests and diseases. So we're going to watch a short video on soil structure. And again, we'll be looking and learning about how bacteria and fungi are some of our biggest helpers in that space.
Why is it important to have well-structured soil? A well-structured soil will allow water to gradually flow through it. As the water passes down through the soil profile, it creates a vacuum, and so fresh air is drawn into the soil, thus creating aerobic or oxygen-rich conditions. This is crucially important, as the majority of disease-causing microbes cannot operate in oxygen-rich environments, and they are forced to shut down. Meanwhile, beneficial microorganisms thrive in oxygen-rich environments. Water droplets are held in the numerous cracks and crevices, and in organic matter for long periods of time, thus increasing drought resistance. Good structure allows plant roots to move effortlessly through the soil profile without having to work hard against compaction layers. In soils where good structure has not been built by beneficial microorganisms, compaction layers are easily formed by vehicles, by human and animal traffic, and where the soil surface is left bare by rainfall. Compaction layers force plant roots to grow sideways, limiting their access to nutrients and water. When plant roots break through compaction layers, they encounter anaerobic or low oxygen conditions. Those are occupied by anaerobic microorganisms, which produce highly acidic compounds. Roots cannot survive in such conditions where the pH can be lower than 4. When water flowing through a soil meets a compaction layer, it can't get through, and so, in heavy rain, this can result in a saturated layer of soil sitting on a hard compaction layer. If there is an incline, the topsoil can start to move, resulting in a landslide. This type of soil erosion is a big problem in many places around the world. So how is good structure built by the soil food web? Well, this depends on the type of soil we're dealing with. The terms sand, silt, and clay describe the size of the soil mineral particles. Sand particles are relatively big, and so are the spaces in between them. Water quickly finds its way through the gaps, and not very much water is retained, especially if the organic matter content is very low, as organic matter can absorb huge amounts of water. So sandy soils tend to be very dry and are prone to wind erosion, which is the process by which the topsoil is blown away and lost. Soil erosion is a serious environmental problem and poses an existential threat to humanity, as we only have around 60 years of topsoil left in the world, according to recent estimates. With the soil food web in place, the picture rapidly changes. Beneficial bacteria produce sticky compounds, or glues, which make it easy for them to adhere to the surfaces of organic matter and sand particles. These particles then adhere to one another, forming microaggregates. Notice how as things clump together, small spaces are created around them. Fungi also produce some glues, and their long, narrow tube-like structures act like ropes, tying together the microaggregates to form macroaggregates. Now even larger spaces are formed in the soil structure. When microarthropods, earthworms, and insects burrow through the soil, they create even larger spaces like tunnels. All the while, the soil food web is increasing soil organic matter in the soil profile by decomposing the surface litter into humus and transporting it deeper and deeper into the soil. Soon you can dig down and discover layer after layer of dark chocolate brown soil, where there was once only lightly colored sand. The result is a structure that will hold water for several months after the rains have stopped, making it available to plants in times of drought. Roots are able to dig much deeper and to access more water and nutrients in parts of the soil that were never available to them before. In essence, the subsoil has now become topsoil. Dr. Ingham and her team have worked on landscapes such as pastures, turfed lawns, and golf courses, where the grass roots were only able to grow an inch or so into the soil because of the presence of compaction layers. In just a few short months after the soil food web is restored, those same roots are able to grow more than 3 feet deep, accessing more water and nutrients than ever before. In clay soils, the problems are a little different. Clay particles are often referred to as platelets, as they are kinda round and flat like the plates you have in your kitchen. Without a balanced soil food web, these platelets get stacked up on top of each other, forming very tightly compacted structures. Compaction layers can form very easily in clay soils. The solution to this problem is finding a way to get those clay particles to stop stacking. This can be achieved by a process called flocculation. Flocculation occurs when sufficient positively charged calcium ions are present on the surface of the platelets. This results in them repelling each other just like magnets do when their poles are aligned. The soil food web increases flocculation, and once this is achieved, structure can be built in just the same way as with sand particles. The result, once again, is a well-structured soil that enables plants to access more water and nutrients than ever before. For farmers, this means increased yields, cost savings due to reduced irrigation requirements, and even further cost savings as there is no longer any need to plow. Lower costs and increased yields result in greater profits. For the environment, there are a number of benefits. A well-structured soil that is home to the soil food web purifies water as it passes through the soil profile. This is because any contaminants in the water are seen by bacteria and fungi as nutrients, and are rapidly absorbed into their bodies as the water passes by. Soil erosion by wind and water are greatly mitigated, offering food security to future generations. And finally, water vapor is one of the most abundant greenhouse gases. By enabling more water to infiltrate the world's soils and to flow into aquifers, we may be able to contribute to the fight against climate change. For more information about the numerous benefits of the soil food web and how you can get involved, visit soilfoodweb.com.
All right, so we learned a little bit more about soil structure and how the microbiology plays a significant role. And Elaine, I think we have a slide for you to be able to talk through. You're on mute, Elaine, just to let you know.
There was too much noise in the background here. Sorry. So talking about soil structure, I touched on it briefly, but we have to rebuild that structure. As soon as you till anything, you're slicing and dicing all of these beneficial organisms, except for probably the bacteria. Certainly you destroy the microaggregates present in the soil when you till, or you break up a large proportion of them, which means the bacteria have to get back to work. And if you've killed a number of those bacteria off by squishing them between particles as you tilled, as the bottom layer of that tillage equipment pushed against the soil, compacting the soil as you're driving along, that means you don't have as many bacteria, and so the recovery is slowed. You don't have any of the fungi left because most of them were sliced and diced and crushed. Certainly, if the farmer's using inorganic fertilizers, because the bacteria and fungi aren't really doing their job, the protozoa, the nematodes, the microarthropods were killed because of the tillage problem. They put out the inorganic fertilizer because the plants aren't doing so well. The plants start to grow using that inorganic fertilizer, telling you that you are not, you don't have a good healthy food web in there, because if the good healthy food web was present and you applied inorganic fertilizers, the plant would not increase its growth rate at all. So the very fact that your plant responds to the input of those inorganic fertilizers tells you that there's something wrong with your food web and you need to fix it, or you're going to be stuck putting out inorganic fertilizers. And as soon as you put out inorganic fertilizers, you've imbalanced what your plant is taking up. And so now that plant is not capable of resisting insect pests or other problem organisms. And so now you're dealing with the requirement to put out pesticides of some kind, fungicides, nematicides, or herbicides, all of those things with the weeds. If you go far enough backwards in succession, you're going to have mostly weeds come up. And so all the weed seeds in your soil bank are going to germinate and grow. How do you prevent that? And that's all about the balance between bacteria and fungi. There's some really interesting work going on at the University of Tokyo in Japan, where they are showing that if your soils have the same amount of nitrogen, but that nitrogen is present as nitrate, you're going to grow an immense number of weeds. If you reduce the amount of nitrogen from nitrate, and you now have an equal amount of ammonium present, you will significantly deplete the sets of seeds that are going to germinate and grow. So you've reduced over half of your weed problem. What if you could apply, you could get all of that nitrogen present as ammonium? None of the weed seeds germinated, none of the weed seeds survived. So we want to get that fungal biomass up and going as rapidly as possible, because it's what fungi do is release ammonium from their biomass, and then they make certain that that ammonium stays as NH4, because fungi produce slightly acidic organic acids, and they maintain pH down around 6.4 to 6.5. If you've got just bacteria, you're not going to be building structure. It's going to be very easy for your soil to compact. And then you have the production of some significantly low pH organic acids down in the 2s and the 3s. And so balancing, mixing that with the existing soil, you end up something, with a pH between 3.5 and 5.5. So these are just some of, I can't go through all of the important things that are improved by forming soil structure, by getting the bacteria in to build the microaggregates, the fungi back to build the macroaggregates, so you get really good spaces for oxygen to move through. Water will be held in the pores, so you don't have to irrigate most of the summer. Maybe if you've gotten really bad drought periods for years and years, you might end up having to irrigate, but it is not something you're going to have to be doing year after year. It's kind of like, how did farmers back in the 1700s or the 1800s, how is it that they could be growing good crops year after year after year? Well, they used a lot of compost, and they did not go out and till the way that we do. We don't, when you have to be behind a bull or behind a horse or a mule or donkey, whatever, and you're trying to keep that animal going straight along the plow line, and you are providing the weight to keep that plow down at the same depth all the way through your field, you can't do very much as a farmer. You can't be, you can't own 1,000 acres farm. So, the size of the farms tended to be around 20 to 30 acres, and that's all that a human being could manage. So you only tilled once a year. You didn't till every time you had a weed problems, because you just didn't have the manpower to deal with that. So roots always grow deeper. People often ask me, how deep can roots grow? Well, it depends on what plant you're looking at. But when you're looking at like a Douglas fir, I have had many people in forest science tell me that root systems of Douglas fir go down, maybe 3 to 5 feet, and then they go sideways. That is abnormal. That is a plant that's not going to be healthy. It's going to be diseased. It's got problems. And it's because of compaction in the soil. When they went and clear cut and, all that traffic across the soil compacted the soil, rain on bare soil compacts. And so you end up with a situation where you plant your little seedlings, their root systems get down to that 3 or 4 foot mark, or not even that deep perhaps, and now the roots go sideways. And we're not going to have healthy forests. So you've got to think about all these factors going to develop the important characteristics of good, healthy soil, which is plenty of oxygen, plenty of microorganisms. You don't have the diseases and pests. You don't have the weeds. So all of that together, you end up not having to work that hard to grow a bumper crop of whatever your chosen crop is. So we like to teach you how to do that. Over to you, or back to you, Brian.
Yeah, back to me. And I think we got a question here that just kind of fits right into what you were just talking about. And it's from Laurie, which is, can you talk about the problem with tilling? Which I think, we've hit pretty hard, but this is a question I get all the time too. Is hand tilling the garden okay? Is mechanized tiller literally cutting microorganisms in half? I think by hand tilling, I think you mean like a broad fork or something like that.
Yeah, a broad fork, because when you put the broad fork in and just go back and forth, you're mostly just cracking the soil. You are not, mixing every part of that soil and disturbing it and forcing the structure within that soil to fall apart. If you want to do some simple comparisons, look at your soil before you till it, and you realize that there's not much structure there to begin with. What does it look like after you've tilled it? And it's now all of even the small air spaces, the small passageways have collapsed. When you shake that soil, how much of that soil just slides off your hand because nothing's holding it. So that's the difference. Really good structurally developed soil, when you pick up that handful, if you're lucky enough to be able to pick it up, because the fungi might be tying it all together and it doesn't want to let loose. But if you can pick it up, shake it a little bit, and nothing slides off. All of the soil particles are bound by something keeping them in place. If you have a heavy rainfall on soil that's not well structured, anywhere there's compaction, the water's going to back up and run downhill along with some of your soil and all of your soluble nutrients. If the water can infiltrate a little bit, it's going to get down a little bit deeper, but then it still has the same problem. No structure. Your soil's going to go anaerobic with not pleasant consequences for the growth of your plant. Or the leachate's going to move downhill, and you're fertilizing your rivers and lakes and streams, causing damage all the way to the ocean. And then you're still causing damage to the coral reefs, to those protections of our shores, of our continent. So we need to start getting these organisms back into the soil to reverse the damage that's been done. None of you are blamed for the damage that's happened. You're doing your best to understand all of this and your best to reverse the problem. So it's you're to be congratulated for coming to these kinds of talks.
I just want to mention on the broad fork, I even look at the broad fork as probably still a transitional tool as well. I mean, once you are in the process of trying to re-establish a good soil food web and build structure in your soil, you might need a broad fork for a few years just to help that process along. But eventually your beds, like at my homestead here, my beds are so well structured, I wouldn't even think of using a broad fork. There's no need to. I mean, I can literally go down with my hands and plant all my transplants because it's the soil so well structured. Just push it apart, put yourself to that space. Any other comment from Adrienne or Wes? Wes, you're on mute, just so you know.
Hate it when I do that.
Yeah.
It's been quite a few times that I've received samples from clients for analysis, and you look at it and you see right off the bat, yeah, there was a lot of tilling going on. Like I've received samples that are like, I kid you not, like a clump of modeling clay. That's how compact it was, no air in there at all. That's just nothing but tiny clay particles packed tightly together. And lo and behold, yeah, there's a few bacteria in there, but not much more. And it's, you can tell right off what it's going to turn out to be. And there it is. There's just not much biology.
Adrienne?
Absolutely. I think the best use of something like a broad fork or these other tools or hand tilling, whatever it is that, breaks up the soil, the best use of that is to enable those microbes to get down in there, right? So whether it's an injector or even using water knows where to go as well. So using water to flush the system and then follow that with a flush of one of our liquid extracts that you saw in that initial video. We can get those microbes deeper into the soil, which can allow those processes to infiltrate deeper and allow more life to fill in the surface area of the soil.
Yeah. And another thing that it seems like a misnomer for a lot of people, but there are many cases where I will recommend to a client that we need to go in and actually rip that soil. We need to use a subsoiler or a keyline plow. And it's like, what are you doing? Well, the soil is already denuded of the microorganisms. It's so heavily compacted that it's going to take quite a bit of time to allow for the natural systems to help break down that compaction. where we can do one last time where we'll go in with a subsoiler or a keyline plow. But the key to this is if you're going to do that work, then make sure you add the more microorganisms at that time frame. So either with top dressing with compost and incorporate it into the soil, or applying a compost extract at the time that you're doing that subsoiling or that ripping. And then from there on out, it's like, look, farmer, that's it. That was our last go. We broke up the compaction. From here on out, now we're going to let the natural systems try to rebuild the structure in that soil.
Yep. And then when we're planting, also, we want to think about in that furrow, when we're going to drop that seed onto the surface of the soil that you just opened up by making that furrow, having soaked the seed in the proper mixture of microorganisms, or laying down a little dribble of that same liquefied compost material, we can make certain that we're getting those microorganisms right around the root system of that plant, so everything gets inoculated really well.
Okay, I think we take one more question on soil structure, then we'll move on to pests and diseases. This question is from Nikki, and Nikki says, I have just had my property landscaped and the soil was compacted and has been further compacted by the heavy machines. Is there a way to ensure that the soil food web can be reestablished? The soil in my gardens currently hold a lot of moisture, and I'm concerned that plant roots won't have what they need to thrive.
Yeah, the problem is they've left you with some serious problems that, it would have been nice for you to be able to go out to them a couple months ago and say, I want you to till everything up, and then I don't want to see you driving across my lawn anymore because you're just compacting the living daylights out of it. So you're probably going to have to go in and do some injection in your lawn so that you're getting the biology below that compaction layer. So figure out where your compaction layer is, how deep it is. We typically use something like penetrometers, or we'll go in with a trowel or something and try to figure out how deep down that compaction layer goes. And then that tells you how deep you're going to have to inject the organisms into the soil and get them reestablished. In your beds, same kind of thing. How much compaction is there? And the easiest way to deal with that is to use a penetrometer. And you might not know what a penetrometer is, so I'll explain. Basically, it's a metal rod about a quarter of an inch or half inch steel rod. You sharpen the one end that's going to go down into the soil, and then you get a pressure gauge where the pressure plate is on the bottom of that gauge, and then you start pushing on the gauge so you can see how much effort it is for you to push that metal rod into the ground. And if you're above 150 psi, grasses and most of your veggies, most of the things that you would be growing in your beds, your flowers, your roses, cannot push their root systems through a soil that's compacted more than 150 pounds per square inch psi. Some plants produce tubers, they've got water roots, and they'll push that through 150, but they can't get any further than 300 psi. So you want to be pushing that metal rod in and finding out where you've got the bad spots. And hopefully there's some good spots in your lawn and in your beds. But then this helps you learn, helps you find out where you're going to have to concentrate your efforts to get that compaction decompacted.
Yeah, I would second that with using the deep root zone injector. I had a client that she had a beautiful Scots pine. They built their house, and in the process of building their house, they heavily compacted the land around that tree. And sure enough, that tree was really suffering. When I showed up on site, you could tell the branches were wilting down, and it was being infested with turpentine beetle. And turpentine beetle will burrow through the bark and get into the cambium and really play havoc with the tree. And the way the tree protects itself is by pushing out lots of sap. and this tree did not have enough energy to even make sap anymore to try to combat against the turpentine beetle. And we went to that tree and found it was compacted, used a penetrometer, and it was significantly compacted. In fact, a lot of the roots at this point had just been laying on the surface of the soil. You could just see, 10, 20 feet away from the tree, there were still just roots just laying on the surface. We went in and drilled about 30, maybe 40 holes using the deep root zone injector and added in compost extract. And really, we punched through that compacted zone. Within 2 weeks, that plant went from drooping to now needles moving upwards, and then the tree just started pushing out tremendous amounts of sap. And it was that fast a response to be able to allow those roots to now grow into new areas and get to uncompacted soil. And boy, it was significant, the difference. So it definitely works very, very well. Wes, Adrienne, anything else you want to comment on this?
That's an awesome story. I really like that one. Thank you.
I do too. Yeah, I want pictures to go along with it.
Oh yeah, I do. I got some pictures. Yeah, it was amazing how much it and now the tree is just healthy as all get out. It's very, very happy tree.
Next promotion.
Okay.
Want to see that?
There you go.
Yeah.
There you go. Pictures or it didn't happen. All right. Let's move on to the next section here, and we're going to be talking about inhibiting pests and diseases. So we're going to watch a short video on that.
So, how does the soil food web inhibit pests and diseases? Well, if you've watched the animation on building soil structure, then you'll know that a well-structured soil is a hostile environment for most disease-causing microorganisms. That's because these anaerobic organisms don't do well in an oxygen-rich environment. This means that they are easily outcompeted by the beneficial aerobic microorganisms that do thrive in oxygen-rich environments. Access to foods for the anaerobic microorganisms is severely limited by the competition from the beneficial aerobic microorganisms, making life even more difficult. The sugars and carbs that are put out by the plant are released from the roots in order to feed beneficial microorganisms, as explained in the animation on nutrient cycling. Naturally, these beneficial organisms congregate around the roots. In fact, they totally cover the surfaces of the roots in order to get immediate access to this valuable food source, thus making it very difficult for anaerobic organisms to access the food supplied by plants. As the surface of the roots are covered by beneficial organisms, the microscopic infection sites which the disease-causing organisms are looking for cannot be accessed. So the plant roots are very well protected against infection. But what about the above-ground part of the plant? Well, it just so happens that plants release sugars and carbs from their leaves too, and from their stems and fruit. In fact, from all parts of the living plant. And the beneficial microorganisms can be found there too, feeding on the compounds being released by the plant and providing protection to the infection sites in return. Here's an interesting thought. Imagine picking an apple from a tree, and the surface of that apple being completely covered in beneficial microorganisms. What effect do you think nature's little defense force might have on your gut biome? So, without access to food and facing oxygen-rich soil conditions, the bad guys are severely weakened. Many of them are consumed by aerobic predators. Insect pests can also be a problem for farmers. These are much larger than the disease-causing microorganisms we just looked at. But real-world trials have shown that with a balanced soil food web in place, insect pests are deterred from attacking the healthy plants that are grown. Healthy plants produce chemicals that deter pests, whilst plants that are stressed are less able to do this, and so they're more susceptible to attack. Having the soil food web in place results in cost savings for farmers, as they no longer need to spend time and money spraying pesticides. And they also see their yields increase, as less plants are attacked. According to the UN, pesticides are largely to blame for the decimation of insect populations by approximately 25% per decade over the last 30 years. Bird populations are also crashing as a consequence. There is mounting evidence that the human biome is one of the most important factors in human health. Could eating food grown in natural soils be good for our health? We at the Soil Food Web School think so. For more information about the numerous benefits of the soil food web and how you can get involved, visit soilfoodweb.com.
All right. I think, Elaine, we've got a slide for you to be able to talk to.
Okay. I'm dealing with pests and diseases. So
Yes.
one of the most important things is to remember aerobic conditions. Very few of the bad disease-causing organisms or pests survive in aerobic conditions. Most of them, I mean, you can find a few species, but they're not typically the ones that are going to decimate your crop. The ones that really decimate are the ones that require anaerobic conditions, which means there has to be something preventing oxygen from getting into that soil, or the aerobic organisms would go take over that environment. So what you have to figure out is what's causing the anaerobic conditions so that we can get rid of that problem. So we want to make certain that we're doing the things to maintain aerobic conditions so that the roots can put the roots as deep as possible into the soil, feed all of the bacteria and fungi, tell those bacteria and fungi what the nutrient is the plant requires. Bacteria and fungi make the enzymes to pull those nutrients out of the organic matter, out of the sand, silt, clays, rocks, pebbles, etc., inside the bodies, and then the predators eat the bacteria and fungi, releasing those nutrients to the plant, so the plant can take up every bit of nutrient that it needs. Now, a lot of the time we see excess nutrients being produced because the protozoa, the nematodes, the microarthropods, the earthworms come and eat those bacteria and fungi, releasing those nutrients, even more nutrients than the plant requires. So what happens to those nutrients not taken up by the plant? Well, because the predators didn't eat all the bacteria, didn't eat all of the fungi, and so those bacteria and fungi that didn't get eaten take up those excess soluble nutrients and store them back in the pantry till we go through that cycle over and over and over again. And the bacteria and fungi always stay ahead of their predators because they're happily building, the bacteria and fungi are happily building microaggregates, which will protect them against some of the predators, macroaggregates that will protect them even more from those predators. So there's always a place, a niche for these organisms to escape predation. And so the nutrient cycling goes on and on. So that part of mitigating diseases is really because we're feeding the plant. So it is never stressed. It will always be resilient and be able to resist those diseases and pests. Now, in addition, of course, in the soil and on the bark, on the leaves, on the surfaces of the flowers, on the surfaces of the fruit or the seeds that are being produced, we've got competitive microorganisms whose favorite food are the diseases and the pests, the problems. So we want to make certain we've got these competitors that will go out and eat all of the pest species, the fungi and the bacteria and protozoa, whatever bad guy nematodes or root feeding nematodes, whatever above ground insect pests, whatever. We want to make certain that we have put back in the competitors. So we want to be getting the microarthropods. We want to get the nematodes that attack and consume the root feeding nematodes. You know, a lot of people think that there's only bad guy nematodes. That's what you've been led to believe by chemical companies that want to sell you a nematicide that you don't need. Make certain that you're not applying a nematicide when it's just good guy nematodes present in your soil. And most of the chemical nematicides that we have are no longer very effective at taking care of the disease of the root feeding nematodes. So what's the better approach to deal with the root feeding nematodes? Well, it's to put in all of the different kinds of nematodes, because the bacterial feeders and the fungal feeders are bright enough to understand that if a root feeding nematode comes along and starts attacking their root system, that the bacterial feeder and the fungal feeder is going to be harmed ultimately by the result of that root feeding nematode attacking that root system. And so they're going to push those root feeding nematodes aside. They're going to make certain that those bad guy nematodes get introduced to the predatory nematodes as rapidly as possible. So there's mechanisms in the soil that we're just beginning to understand, and how complex and nature's been at this process of dealing with how to take care of plants. And obviously she's been pretty good at protecting the plants that she wants to grow in this place at this time. So for the last, what has it been? 3.5 billion years? The bacteria and fungi, protozoa have started to develop on this planet. And so they've been here working happily away and figuring this all out. So it works very well. We want to have those microorganisms that make inhibitory compounds. But unlike humans, where we take an organism that makes an antimicrobial, it makes an antibiotic, we grow those bacteria out to billions and billions and billions in huge amounts of pots, and we force those bacteria to make those antimicrobial agents. So we've got concentrations of the antimicrobial or compound, just huge, way beyond, way beyond normal concentrations. When we're working in the soil, we don't want abnormally high concentrations, because those high concentrations kill all kinds of other things that are beneficial. So we want to make certain those organisms that make the antimicrobial products are present in normal concentrations around that root system. They're interacting in normal ways with the other bacteria, fungi, protozoa, nematodes, microarthropods, earthworms, and all those guys. So the inhibitory compounds that are made are only local, only where the disease-causing organism is trying to cause a problem. Make sure that we've got those bacterial fungal species that will be able to make those inhibitory compounds. We then, we want competition, so we want to make certain that our leaves are covered by surfaces. Those surfaces are covered by organism that if the spore or the disease-causing organism falls on that leaf surface, it's not going to be anywhere near the surface of that plant itself. What if that spore does manage to germinate and that spore starts putting its hypha out? We want those things that eat fungi, that eat the bad guys. We want them present in our inoculum that we spray, the compost extract, compost tea that we spray out on the leaf surfaces, so that even though that spore has started to germinate, somebody's going to go, aha, that looks like dessert to me, and goes out and eats it. So competition, inhibition, and consumption. If you're, I can go on and on. So I think I've had enough to say so far. So I'm going to let everybody else have a chance.
Sure. In fact, we've got a question here I think fits really, really well with what you're just talking about from Alessio. And the question is, aren't diseases and pests part of the soil food web? In fact, they exist in wild forests. So how might we make sure that we don't create the good conditions for another disease or pest to grow and compromise our harvest? For me, I think that's just a matter of perspective. We want to have pests and diseases in a natural ecosystem. They have a very significant ecological role. It's not like we want to get rid of all pests and diseases. Then we wouldn't have the ability for good genetic lines, because that's one of the significant things that they provide to us is to take out weak organisms. We just want to make sure that we have the conditions so that we don't have the pests and diseases thrive, and that's like building good soil structure and things like that.
Right, if I were to quote Vandana Shiva, she says that insects are not pests, fungi are not diseases, but when they are allowed to thrive in a habitat that doesn't have a healthy community ecology, then they can act in a way that is unhealthy for the food that nourishes us. So, yeah, thinking about these pests as normal community members when they're in their normal abundance numbers, they're totally fine. A few nibbles here and there is not going to knock out a crop. However, keeping those populations in check is key, and that is the key that the soil food web can help contribute to.
Agreed. Elaine, anything? Wes? Wes, I think you're on mute.
Loves that mute.
Hard.
I was just thinking it helps to just consider pests as, when they attack a plant, they're doing exactly what they're supposed to do, because, going back to succession, that plant is not growing where it's supposed to grow, ecologically speaking. So the pests, of course, they come in and they remove it. That's what they do. And so this all kind of makes me think of, you're not only using a chemical to destroy a pest, like a nematicide to destroy root-feeding nematodes and other nematodes if they happen to be there, but here in California, there are places where soil fumigation is used to get rid of nematode problems. And so in a case like that, you're taking a chemical and killing everything. The idea is to sterilize the soil. So you're creating a blank slate for anything to move in. And the first thing that's going to move in is the pest that's going to take out that plant, because the successional level of that soil now is completely way down in the beginning, early successional. And so, yeah, I suppose it just helps to think of pests as they're just doing what they're supposed to do. So, we need to create the conditions that make it so that they're not needed. The pests don't come in and say, oh, there's a job for us to do here, because those conditions don't exist.
Yeah, I kind of think of sometimes pest diseases almost being our allies in a weird way, in the sense that they give us information.
Mm-hmm.
If all of a sudden you have a pest or disease outbreak, it's telling me something that, okay, something's not healthy in this ecosystem. We need to be able to go figure out what that is and how to rectify that.
Yeah, we got to fix it. So what's the fix that you have to apply? That's what the disease or the pest is telling you about. There's not enough airflow through this vineyard. And so the vines are being attacked by a particular problem organism. It's sometimes difficult to figure out exactly what the message is from Mother Nature when the disease outbreak starts to happen. But that's why you want to be out walking through your agricultural fields, so that you find those places just as the problem starts to announce the fact that there's something wrong here and you better pay attention. Because if you ignore Mother Nature's messages, she's just going to send a nastier message. And if you ignore that message, she's just going to send something even nastier. So want to make sure that we pay attention to the information Mother Nature's trying to tell us.
Yeah,
and there's a lot of really cool predators out there. This consumption element of how the soil food web contributes to protecting plants. Parasitoid wasps will come and lay their eggs inside of caterpillar, develop inside that caterpillar, and spring out and go help maintain those control the population. It's beautiful. I mean, it literally is beautiful, but also is a little the stuff sci-fi is made of.
Alien.
Well, I think, also in a lot of current practices, we're so heavy-handed. You see a pest, I get my miticide out, I get my nematicide out, and you nuke really anything that it could be as a beneficial stance, and then we're kind of resetting ourselves. One of my farmers, I was really proud. We've been in a block where we've been, only applying biology, and his other blocks he's doing conventionally. And we got a little bit of spider mite pressure coming into that block. And he's like, I want to wait and see, how much the pressure increases. And all I had to do was just wait a few days, and then predator mites came into the situation and really controlled the spider mite population. So without being heavy-handed and knowing that we were building an environment that was going to foster both the predators as well as the harmful organisms, when you get in that balance, then it works really well. Here in my homestead, I see some aphids starting to pop up somewhere. I don't worry about it. I know for a fact within a few days, the predators of aphids are going to be all over that, and they control it. And it becomes kind of a comforting thing. Before I used to freak out. Oh my gosh, I got this. Oh my gosh, I got that. But now it's just kind of an observational thing. Oh, there's some aphids. Walk by it. I know in a few more days. Oh, there's more aphids.
They should be
works
there.
really, really well.
They really should be there. They shouldn't overpopulate, but they should be there.
Totally.
Aphids can be a really useful source of honeydew that can feed some of those parasitoid wasps that can also possibly control your caterpillar outbreak.
Exactly.
Also get to know get to know those predators of the aphids. Know what a lacewing looks like. Know what a syrphid fly looks like as a larva. It looks kind of like a caterpillar. Learn what that little Rice Krispie aphid looks like when it's, puffed up, because that's actually a mummy that has another parasitoid wasp growing up inside of that aphid. Back to you, Brian. What were you saying?
Well, no, and that's the thing is when you're in a healthy ecosystem, and I have a few, you want to feed your predators. They need to have a food source to be able to stick around. And the aphid damage is so insignificant to the production of that plant. I'm not worried about it. I'm not getting a crop from those plants that I'm growing. And that works out really well. That's that whole balance that you're shooting for. I mean, it's neat once I learned that, lacewings, they are very nocturnal. And at nighttime, I can go out and I have an oleander plant right out by my back door. And at nighttime, I was out there and there's hundreds of lacewings flying around. They're laying eggs. And then the next day, there are a whole bunch of lacewing eggs underneath that oleander. And I was going to trim that oleander, and it said, "No, Mom, I'm going to wait till the lacewing eggs hatch, and then I'll go ahead and trim it back." But it's just becoming more, I think, educated about that, and not just going to the quick trigger. Oh, I got a problem. Let me go get a chemical to go fix that.
Nuke it.
Now, granted,
kill it.
If you don't have an ecosystem and your soil is poor and your whole environment's damaged, yeah, we got to make that change, So sometimes people will still go use those triggers, but we've got to start. We got to make that switch.
Yep, one plant's herbivores, another plant's pollinator.
Exactly. I say we take one more question, and this question is an interesting one, and something that I think is becoming a very common problem, which is this one's from Allison. Would love to hear an ecological response to Asian jumping worms. Just learned about them from my own nursery and see their impact on forest cover plus increasing soil erosion. What is your perspective on them? And this is going to be kind of that non-native invasive kind of question, I think.
Yeah, I know we worked on that, and we have a whole write-up on that in the foundation forum. I don't remember right now off the top of my head what we said was going to be it should be the way to deal with the jumping worm. Can you remember, Brian?
I don't remember at the top of my head, but I think the thing that clicked for me on this question was saying that, as we live in this kind of global community, and there's so much movement of people and things that, pests and diseases become taxi cabs or get taxi cabbed into your environment. And it is a problem because, you'll see from some non-native invasives is that there's no natural competitor or predator for them. So it's like, ooh, nobody eats me. Guess what? I don't have any controls on my production or my population, and they'll explode. And so, there are biological controls that we can try to use. Some of the, like a navel orange worm, there's different nematodes that would do a very effective job. So I think for those non-native invasives, we got to try to find some biological things that we can do to try to help control some of those populations, while an evolutionary type of thing occurs where now predators start to, okay, I can eat Asian jumping worms or whatever it's going to be. It's going to be a challenge, I think, moving forward for sure, the more that globalization kind of occurs.
Maybe we could get less stupid about carrying samples of materials across bioregions. Keep it in the bioregions. Don't be, is there hope for humanity? Let's hope.
I think so.
When we think about compost as a source of microbes, it's also important to think about the boundaries of, if you are producing compost out there, think about how far is responsible for that to be shipped, partly for the organism's sake, because they don't like being in closed containers and shipping boxes, but also for the sake of where those microbes might end up.
Yeah, you take New York organisms and you ship them out to Arizona and spread that compost out, and then wonder why nothing good happened. Well, those are organisms that like rain every day, or at least once every 3 days, something like that. They don't do well in 105-degree temperatures. So it's not the right habitat to expect those organisms in that compost from New York to deal with. And it's kind of vice versa the other way around. I wouldn't take something from New Mexico and transport it to South Carolina, because that's absolutely not the environment that those organisms in that compost are adapted to. They're just gonna probably die. And so waste of money. Go within your bioregion to buy really good compost and be looking for the label of biocomplete. Then you know that all of those organisms are in that compost to a minimum amount that you need to have.
Which means we need more folks making biocomplete compost out there.
We do.
Yep, and once you start making really good biocomplete compost, you can increase that price quite a bit. People realize almost right away that it's worth the money.
Get much more bang for your buck.
Yep.
Especially for the work we do. When I evaluate compost, I look to see, well, how much compost do I need to use? And in some cases, it's so poor that the amount of compost, of poor quality compost, is 10 times, 20 times, 30 times more than if I had a really good biologically complete compost to be able to use. So, okay, I think we're at the top of the hour. Any closing comments before we move on to just talk about our future promotions or webinars? Okay, I think, Adrienne, we can go to the next slide. So I just want to leave everybody with a reminder that we've got 3 more webinars as part of our October promotion here. Again, webinar 2 is going to be proof that regenerative ag works, and it's going to be on October 13th at 10 a.m. Pacific timeframe. And then webinar 3 is going to be how to rapidly regenerate your soil, and that's going to be on October 21st at 11 a.m. And then we have webinar 4, which is going to be Meet the Soil Regen Pros on October 28th at 2 p.m. Pacific time frame. So hope to see you guys there. And there's a lot of good differing topics. So, I encourage everybody to go out to all 4 of these because you'll get quite a bit out of being able to listen into those webinars. And at this point, I'd like to give some thanks to the people working behind the scenes. We have a lot of folks that work really, really hard to put on these webinars, and they're keeping us on track, getting the questions organized for us. So I want to thank everybody that's behind the scenes supporting us. That is fantastic. And I'd like to thank our panelists today. Obviously, Elaine, you bring so much to the work that we do, and really just being a beacon of information and support for us as we start to do this work. So thank you so much, Elaine. And then I'd like to thank Adrienne and Wes for being panelists in today's and providing really good commentary and really making it an interesting webinar.
Thank you so much, Brian.
Thank you.
And thank you, Brian.
Great hosting job.
All right. Okay, so at this point, the webinar is going to conclude, and we hope to see you guys in October 13th. Bye, everybody.
Ciao.
Bye.